I hope you have seen my video. Introduction to Biochemistry. In this video, I will focus on lipid function. Lipids are most recognized as fats and oils. Waxes are also lipids. Lipids have two major functions: energy storage and construction material for cell membranes. Long-term energy storage in the body is done by lipids, usually in the form of triglyceride molecules. In mammals, lipid storage is done in specialized fat cells of two types: brown fat cells and white fat cells. Fat cells are also called adipose cells or adipocytes. The corresponding fat cell-containing tissues are called brown adipose tissues and white adipose tissues. The difference between brown fat cells and white fat cells is that in white fat cells, the fat is stored as a single droplet of lipid oil, and in brown fat cells, the fat is stored in many smaller droplets of lipid oil. The single lipid droplet in white fat cells takes up so much of the volume of the cell that the nucleus of the white fat cell is squeezed over against the cell membrane. Brown fat cells have many more iron-rich mitochondria than white fat cells, and it is the iron that gives the brown fat cells their brown color. White fat cells can return the energy they have stored as triglycerides back to the body by breaking down the triglycerides back into their component glycerol and fatty acid molecules and releasing these simpler molecules back into the bloodstream, where they can be absorbed by other body cells in need of energy. Brown fat cells cannot return their store of triglycerides back to the bloodstream. Brown fat cells burn their triglycerides in their many mitochondria to generate heat. Mammals, and even humans, who live in cold climates, have an excess of brown fat cells to keep warm by burning the fat reserves stored in their brown fat cells. Babies are born with high numbers of brown fat cells. Babies need the heat released from their brown fat cells, as they are not able to keep warm by moving their muscles or to move to a warmer place on their own. As people age, the ratio of brown fat cells to white fat cell falls considerably. Older adults who exercise regularly can maintain a higher ratio of brown to white fat cells. White adipose tissue is generally found concentrated in two places. White adipose tissue is found in the abdominal cavity, viscerally, meaning attached to organs, intestines, heart, liver, etc., and subcutaneously beneath the skin. The subcutaneous fat cells are found distributed individually directly beneath the skin. Brown fat cells are found concentrated in the neck, between the shoulder blades and down the spinal column. Brown fat cells can also be found interspersed in concentrations of white fat cells, coloring it beige. When food is digested, the triglycerides in the food are broken down by enzymes into glycerin and fatty acids. These small molecules are free to be transported through intestinal walls out the digestive tract. Before they are released into the bloodstream, they are reassembled as triglycerides and packed into bodies called chylomicrons. The chylomicrons carry diglycerides, cholesterol, and other apolipoproteins into the bloodstream, either to the liver for further processing or directly to fat cells where they can be stored. The presence of glucose and fatty acid in the blood triggers the release of insulin by the pancreas. The presence of insulin in the blood signals the cells to start storing glycogen and fat. Fatty acids are absorbed into fat cells and converted and stored as triglycerides. Excess glucose in the bloodstream is absorbed by the liver or muscle cells and stitched together by glycosidic bonds to form glycogen by a process called glycogenesis. A 170-pound human of average build could store 400 grams, or 1 pound, or 1,600 calories of glycogen in his muscle cells, and another 100 grams, or 400 calories of glycogen in his liver. Once the stores of glycogen are filled in the muscles and liver, excess glucose is either excreted by the kidneys or absorbed into the fat cells for conversion into triglycerides in a process called lipogenesis. The process of conversion of glucose into fat starts with glycolysis and the pyruvate metabolism of glucose into a coenzyme called acetyl-CoA. Once the glucose is converted into acetyl-CoA, the lipogenesis process continues through malonyl-CoA through a fatty acid cycle to end up with free fatty acids. The glycerol and fatty acid generated in lipogenesis can be combined into triglycerides that can be stored in the fat cells. This whole lipogenic process can go from glucose to triglycerides within the cytoplasm or mitochondria of the fat cell. Fat and triglycerides are only brought out of fat cell storage when glucose levels are too low or in emergency situations. The pancreas can detect the glucose level. When the glucose level falls too low, the pancreas releases glucagon, a hormone that has the opposite effect of insulin. In emergency situations, as a result of stress or fright, the adrenal glands release adrenaline, also called epinephrine. The result of the presence of glucagon or epinephrine is that white fat cells produce the enzyme lipase, which breaks down by hydrolysis its store of triglycerides into glycerol and free fatty acids. The free fatty acids are bound to albumin globular proteins before release into the bloodstream. The glycerol and albumin-bound fatty acids are released directly into the bloodstream, where they can be absorbed by any cell in the body to be used by their mitochondria to create energy in its most usable form, ATP or adenosine triphosphate. If the glycerol and fatty acids are absorbed by liver cells, they are further broken down in a process called ketogenesis. The results of ketogenesis are ketones, acetoacetate, acetone, and beta-hydroxybutyrate. These ketone molecules can be used specifically in brain, heart, and skeletal muscle mitochondria in an emergency situation to rapidly produce ATP fuel so that they can act in emergency cases when glycogen and glucose levels are too low or absent. Besides energy storage, lipids also have important functions in cell membranes. Lipids make good cell membrane materials because they are hydrophobic. They curl up naturally into spheres to separate themselves from water. Lipids like phospholipids have a polar head that makes them amphiphilic, and make them especially good as bilayer membranes. Phospholipids have a hydrophilic phosphate head attached to a glycerol, with two hydrophobic fatty acid tails. The structure of phospholipids makes them amphiphilic, hydrophilic at the phosphate head and hydrophobic at the fatty acid tail. Phospholipids are good for making cell walls because they are amphiphilic. What happens when phospholipids are placed in water is that they naturally form fat bubbles. Fatty acid tails face fatty acid tails to avoid the water. The water-loving heads line up to face the water. And so, you get a fat bubble with a phosphate surface facing the water. If some water is trapped inside the fat bubble, you get a double-layer membrane. The outside surface is phosphate heads. The inside surface of the membrane is phosphate heads that face the water. The tails face each other in the center. It is fairly easy to understand how such a structure could become a cell membrane. If a piece of DNA or RNA gets surrounded by the phospholipid membrane, the membrane protects the nucleic acid, and you get a bacteria cell, a life form. Except you don't. Try mixing DNA and phospholipids in a saline solution, and you get nothing. Life doesn't start spontaneously. Sterols are also lipids. The most famous sterol, cholesterol, is necessary to brain function and the building of cell membranes. Cell membranes generally made by amphiphilic phospholipids that act as a barrier to molecules through the membrane. In places there are cholesterol-rich lipid rafts that are not fully understood, but are believed to take part in the transmission of proteins into or out from the cell. This protein transmission is critical to the function of neurons in the brain and may be important to other cells throughout the body. Other functions of cholesterol thought to be important is to make the cell membrane more flexible and fluid. Hepatic cells in our liver manufacture cholesterol from other lipids. This naturally made cholesterol is released into the bloodstream for use by other cells of the body. If too much cholesterol is ingested with food, it interrupts the body's natural formation of cholesterol. The figure shows a lipid raft in a cell membrane with protein valves surrounded by cholesterol. In the graphic, A is inside cell space or cytoplasm. B is the extracellular space. 1 is the regular cell membrane. 2 is the lipid raft. 3 is the raft transmembrane protein. 4 is the non-raft transmembrane protein. 5 is the glycosylation modifications on glycoproteins and glycolipids. 6 is the GPI-anchored protein. 7 is cholesterol. 8 is glycolipid.
Thank you for watching. you enjoyed this video and want to help me make similar content, please like and subscribe.